Okay, I just described the first pathway by which coronavirus can enter a cell. Now I'm going to describe the second pathway. Now recall, in the first pathway, the coronavirus enters the cell by going in right through the plasma membrane of the cell. This pathway is going to be a little bit more complicated, some more steps to it. So we'll start where we left off last time, or we had this in our diagram last time, where we have the S protein of the coronavirus is docked onto the ACE2 protein of the cell. That's the receptor, the lock, if you will, that the key of the coronavirus fits into. Now, instead of the spike being clipped, remember there were scissors outside the cell that could clip this piece off and cause a rearrangement. What's going to happen instead is, in this case, the coronavirus is going to enter into the cell in a bubble, if you will, that we call a vesicle, that buds inwards. So think about this membrane, the plasma membrane, invaginating inwards to make this little container that the coronavirus can sit in. And coronavirus remains bound to ACE2 via the S protein here. That's a process that's known as endocytosis, okay? We're not going to have a quiz here, so you don't have to remember the names, but if you want to impress people at virtual cocktail parties on Zoom, that's the word. Now, that endocytosis process keeps going until this completely seals off and enters the cell, as shown on this diagram here, where we have the virus now inside this vesicle, which is, as I described, kind of like a soap bubble made of the membrane of the cell, and that's called an endosome. And remember, we still have the spike protein bound to ACE2. And if you recall from the prior video, I'm only showing one of each, but there's 60 S proteins per coronavirus, and there's many, many ACE2 proteins on the surface of each cell. Now, how do you get from here inside the cytoplasm of the cell? If you recall, at the plasma membrane, there was a molecular scissors that cut this bond, all right? And that caused the spike to rearrange in the S protein. The same thing happens here, but it requires an additional step. There are molecular scissors in the endosome, but they only can act when the pH of the endosome is acidic, say kind of like vinegar is acidic. So what happens is, if you remember from chemistry, those of you who took chemistry, protons enter into the endosome from the cytoplasm, and those protons are what makes the inside acid. As the inside gets acid, that actually turns on a pair of these molecular scissors that can now clip the bond, that bond there. You get the rearrangement of the S protein, as I showed on the prior video, and when it rearranges, the spike can destabilize the membrane, and that causes the membrane of the vesicle, of the soap bubble, to begin to fuse with the membrane of the virus, with the envelope of the virus, as shown here. So now the RNA of the virus can enter inside the cell. So these are two separate pathways. This process here, acidification of the endosome, getting the protons in to make it more acid inside, more like, say, vinegar inside, that process is blocked by chloroquine, which you may have heard President Trump refer to in some of his daily briefings. And so that would be how chloroquine could potentially act, by blocking this step. But you have to recall that there's potentially two different ways that the virus can get inside cells. And so if this step is blocked, it could still potentially get in by what I showed in the first part. And this is in the end why we do clinical trials, to figure out, would blocking that be enough to stop the process? And, it might be, but we have to do experiments to figure that out. Thanks for your attention in this second video in the series. In the next video, the third video, I'm going to describe how once the coronavirus genome, once the genetic material enters the cell, and I've shown you now how that happens, how can it take over the cell so the cell now becomes a virus factory? Thanks for your attention. Have a good day.